Medicenna is a clinical stage immunotherapy company focused on developing novel, highly selective versions of IL-2, IL-4, and IL-13 superkines, as well as first-in-class empowered superkines. CEO Fahar Merchant is here to tell us about the company and the large portfolio of drugs in the pipeline and imminent release of clinical data. I'm Martin Gagel with Market Radius Research. Please remember, this is neither recommendation nor investment advice. We're here to learn about the company. Fahar, thank you. Thank you for joining us. I've been looking forward to this for a long time. How are you?
Yes, I'm doing very well. Thank you. Great to catching up again. I think we met about a month ago at a
Yeah.
conference or so, but good to see you again. Thank you.
All right. You've got a lot going on in your company, a very science company. So I look forward to you doing a relatively brief overview on the story to give us the key highlights. We're not going to try to get too
Sure.
sciencey on this, but
Okay.
understand what the implications of the science are. So I'm really
Okay.
looking forward to this. And why don't you take it away? Thank you.
Thank you. Okay. So as always, just to make sure that I put out this disclaimer here for the benefit of our listeners, that encourage them to look at our risk profile, etc., on our website. But let's start with the company itself. So, Medicenna, as you said, we are a clinical stage immunotherapy company. The focus is really to transform how we treat patients using approaches that are unique, but at the same time, very compelling based on the data we've generated so far. And just to give you a bit of background in terms of the company, we started the company about 9 years ago, essentially bringing in some assets. The first one that is the most advanced program is called MDNA 55 or bizaxofusp, or for some, they may want to call it BIZA for recurrent glioblastoma. This is a program that's phase 3 ready. We'll talk about it briefly towards the end of this conversation today. And to complement that particular program, we licensed some really exciting intellectual property from a lab at Stanford University. These are based on what we call superkines, and really developed these superkines using technology that became a part of the Nobel Prize in 2018. So really exciting platform that we've got within Medicenna. And as we went public thereafter, we sort of advanced the company into clinical trials with our second program, which is MDNA 11. We'll talk about it today because we have new data coming up in the coming weeks. And this is a program where we've been able to secure a clinical collaboration with Merck. And we did provide some early data of this clinical trial last year at a conference. And then we had an inbound interest from one of the largest institutional investors in the biotech sector, RA Capital, that conducted a $20 million non-brokered financing, which was providing us with really a very strong third-party validation of our programs and our platform, particularly around our superkine assets that we have. And today, RA Capital is the largest institutional shareholder at Medicenna, and we'll talk about MDNA 11 and our next generation program on the bispecific space. Just for the benefit of patients or the listeners here, we are a clinical stage company. By that, I mean we have data now in about 250 patients, with half of them with glioblastoma, the other half with really end-stage solid tumors. So I'll share that with you today, and that is really crucial. So moving on to our pipeline, the program that we are looking to partner is the brain tumor program. This is recurrent glioblastoma, very aggressive end-stage cancer patients. And we are looking to further provide and present some updated clinical data from this trial in a publication, but at the same time are working aggressively on partnering this asset. Today, though, I will talk about MDNA 11. This is our IL-2 superagonist. And this particular program is, just to give you a bit of background, what is MDNA11? What is an IL-2 superagonist? I think what is really important is to demonstrate or to basically leverage our immune system to fight cancer. And what IL-2 does essentially is boost the cancer, our immune system to really fight cancer very effectively. So if you go back to the slide deck, one of the things what we have is really data that we will share on MDNA11 that's being used on its own, but also in combination with Merck's Keytruda. This is sort of a multi-billion-dollar blockbuster that I'll talk about later on. And then more recently, we just announced the use of MDNA11 in a neoadjuvant setting. So what does that mean? Well, neoadjuvant is very early stage. Before the patient even gets surgery, we want to treat patients when they have their immune system, which is much more healthier and therefore more likely to benefit the patient and hopefully the tumor never comes back. And that's the challenge that we have to deal with. So when we are treating patients right now in the MDNA11 clinical trial, we're really treating patients who are end-stage patients who have a poor immune system to begin with. And therefore, the approach here is to leverage that information to see can we get in and treat patients much, much earlier so that the tumor hopefully never comes back after surgery because we have, in essence, somehow vaccinated the patient by using MDNA11. So that's the third program that we've got going. And then back to the pipeline, I'll sort of focus on the fact that we have MDNA 113. This is our bispecific program. There's a lot of excitement around this particular space, and I'll share with you on that as well. So really, we'll talk about MDNA11, MDNA 113, etc., and briefly about Bizaxo. So let's talk about MDNA11. So MDNA11, we've conducted a clinical trial, but just going to basics around MDNA11. MDNA11 is really based on a molecule called IL-2. So IL-2 has been approved actually, nearly 3 decades ago for the treatment of patients with skin cancer and kidney cancer. The challenge with IL-2 is that first and foremost, it doesn't stay in your bloodstream for very long. So patients have to be administered the drug every 8 hours, and therefore not ideal, particularly when patients have to then stay on and get this drug for 5 days in a row. So really, it's 15 infusions for the patients, not convenient. Second thing, which is really the challenge here, is that IL-2 on its own is incredibly toxic. Actually, patients, when they come to the hospital to get treatment with Proleukin, they have to go straight into an intensive care unit because the challenge is the drug might actually kill the patient. So it's a really tough drug to administer, and therefore, not surprisingly, it's not a popular drug that's not being used. So, to one of the things that is important is that IL-2, although it helped a few patients, it did, in some patients, cause what we call long-term cures. And that is important because, what is out there that actually cures patients of cancer? So IL-2 certainly has an exciting opportunity. The problem is not the best molecule. So with the MDNA11, as have others, what you see is there's been multiple efforts by Roche, with Bristol Myers, by Sanofi, multiple transactions, multi-billion-dollar transactions have taken place where they've taken the Proleukin and created a version that stays in the bloodstream longer. But that alone, as you can see, has not helped. All those trials have failed. And we felt that you have to do something very different. And this is where the Superkind platform from Stanford comes into play because not only are we changing the molecules to make it safer, but we're changing the molecule to make it much more powerful and potent. And third is that we are modifying the molecule so it stays in the bloodstream for much, much longer. So you're treating patients once every 2 weeks instead of 3 times a day for 5 days. So these are the key things we've done with Medicenna's drug. As you can see on the extreme right, all the changes we've done are designed to make the drug safer, more effective, etc. So we're combining the art of using albumin to extend the half-life of the drug. As you can see on this particular structure, is we've inserted albumin. And what albumin does, it sort of localizes the drug at the tumor site and also localizes our IL-2 superkine in the lymph nodes. And these lymph nodes is where your immune cells get trained. It's like going to a school to be educated. And that's where the immune cells get educated to attack the tumor. And therefore, really crucial that we are able to do that. And nobody else uses this approach other than Medicenna. So we are doing two things. We're using the best potential IL-2 molecule, but at the same time using albumin to localize the drug. So just to give you an idea
Fahar,
here, yes, go ahead.
sorry, and just to clarify, the IL-2s and the cytokines, they don't actually attack the cancer itself. It's more
No,
about the communication system within the body. A
right,
cancer sort of, they're like, they disrupt the internal communication system and say, hey, we're not nothing bad for you. So the immune system, you can ignore me. And then your drugs actually say, hey, go attack those things. They're the bad guys. And
correct.
it's a more of a communications mechanism. You're not actually
No,
targeting the cancer itself. You're telling the body to target them.
exactly. So we're
Okay.
reusing our existing immune system and leveraging that immune system and using IL-2 to use and sort of deploy the immune system, sort of creating, let's call it this way. We've got the immune system, think of them as soldiers, and we're sort of recruiting those soldiers. We are training those soldiers, and we are training the soldiers to recognize who your enemy is. I think that's the important thing. You want to make sure the immune system is able to recognize who the enemy is and build a really strong army to attack the tumor. So that's the really important thing. Okay. So
Excellent. Thank you.
so one of the approaches has been, in recent decade or so, the immune system has been leveraged by pharma companies like Merck and Bristol Myers. You can see the world's, second best-selling drug. I mean, the Ozempic just, is now ahead, but the second best-selling drug in the world is Keytruda. And Keytruda is being used, the current revenues are just under 30 billion a year for this particular drug. And in end-stage patients, it shrinks tumors in about 15% of the patients, sometimes much, much lower. And this drug, when patients are newly diagnosed, it only helps about 30% of the patients. The remaining 70% of the patients do not benefit from this drug. And same with Bristol Myers drug. It's called Opdivo, and it has the same mechanism, anti-PD-1. And as you can see, sales of about 10 billion here. So these are big blockbuster drugs that are able to help about a third of the patients, the remaining two-thirds not benefiting. And it is these two-third patients that we've been trying to treat in our clinical trial that have failed the checkpoint inhibitor, they're not responding, or simply too advanced for Keytruda or Opdivo to work in these patients. So this is sort of the early data that we have shared. The key thing here is, can we, in these patients, mind you, these are late stage patients who failed multiple therapies, including the blockbuster drugs, can we improve their survival outcomes or even shrink those tumors? And we're already seeing that. About 30% of the patients in the study so far with multiple different tumor types we are seeing tumor shrinkage, in some cases, complete regression of the tumor, and in tumor types such as pancreatic cancer, which you know, and most of us know, is one of the most aggressive tumor types out there. So it's not only shrinking the tumor, but it's sort of keeping the tumor uh, out of being able to progress for an extended duration of time. So the key thing here is it durable? Shrinking the tumor and then the tumor coming back after 4 weeks doesn't really provide much benefit to the patient. But if you're able to shrink the tumor and keep it that way for several months or years, or potentially cure these patients, it's fantastic. It's just, unheard of. And this is where we are beginning to see those kind of data. So let me pick up this one particular patient, this pancreatic cancer patient. This pancreatic cancer patient received surgery. The tumor had metastasized to the liver and then received multiple chemotherapies. And that patient continued to have the tumor progress, advance, and those chemotherapies were toxic. And then eventually the patient got the big $30 billion blockbuster drug Keytruda. And when the patient got Keytruda, the patient's tumor continued to spread and expanded. And this is in the liver, which is really tough to treat. When you have metastasis occurring in the liver, it's a tough one. Patient entered the clinical trial, and the patient, when we did the first scan, we saw the tumor shrank by about 25%. And as we did more, scans on this patient, we saw that the tumor continued to shrink to a point where 8, 9 months later, the patient decided to take a 7-week vacation. Patient went away on vacation, came back, and the tumor that was in the liver had disappeared, but the patient had a new tumor that had showed up. So what they did was did a single cycle of radiation and started the patient back on MDNA 11. And what happened was within 3 months, the liver lesions had gone, the new tumor that had showed up had disappeared. And December 2023, this patient stopped receiving treatment with MDNA 11 and has not received treatment with MDNA 11 or any drug at all since then. And that pancreatic cancer still remains uh, in what we call remission, and there's no sign of the tumor. So that's pretty impressive. Same thing with a melanoma patient. We had this melanoma patient who had failed multiple checkpoint inhibitor therapies. The tumor in this patient started to shrink as soon as we started to treat with MDNA 11, completely disappeared. And then for at least 10 months now, the patient has no sign of the tumor. So this is showing that we are beginning to see these sort of long-term potential cures with MDNA 11 on its own in patients that are end stage. So, the thing is, where do we go from here? So if we go to the next slide, what you are able to also see is that in addition to pancreatic cancer and melanoma patients, for instance, we are seeing that in endometrial cancer patients. So this is a tumor type. The patients that we treated now with obesity on the rise, there's a higher incidence of endometrial cancer. Patients typically will get surgery. After that, they'll get um, chemotherapy. And after chemotherapy, there is a treatment that's been approved recently is using Keytruda, which is that multi-billion-dollar blockbuster drug, plus another chemotherapy. The two together have been approved for use. And basically what happens is that even those patients do not respond. And this is where we treated endometrial cancer patients that failed surgery, chemo, and Keytruda in combination with a chemotherapy drug. And these patients today, when they fail that therapy, they have no standard of care. That is, there are no approved treatment options for these patients. And we've already found that these patients that were in the study received a combination of MDNA 11 with Pembro. 2 out of 4 patients had tumor shrinkage. And this is, really exciting because otherwise these patients would not typically have any other options. So we're seeing in end-stage patients, there's an opportunity with MDNA 11. Just to give you an example of a drug, for instance, that's being used for melanoma, just to give an idea, even if MDNA 11 was being pursued for just melanoma, you can see the market cap of the companies that have melanoma drugs approved. And you can see that in these patients with a 30% response rate, market cap is about 800 million, nearly a billion, prior to some recent changes that have occurred. But with Medicenna, you can see our market cap is substantially lower. The opportunity for MDNA11 is not restricted to just melanoma. As I've seen in pancreatic cancer, in other tumor types, including endometrial cancer, and you can see the current clinical trial is enrolling patients with these other tumor types that are biomarker driven, where the number of patients is much, much larger than melanoma, which is just 7 to 10,000 patients. So in summary, what we are saying is really that if you look at patients who failed these multi-billion mega blockbuster drugs and are responding to MDNA11 either on its own or in combination, there is a huge market opportunity for MDNA11 with these different tumor types. And we think that if you take this drug earlier stage in patients who have just been diagnosed with cancer, where the patient has a much better immune system, we think that this will be really a groundbreaking, paradigm-shifting treatment option for the patients. And this is where the NeoSite trial, this is the slide that you can see. This is funded by the Melanoma Foundation in Italy. Medicenna's requirement is to help with the regulatory filings, but also provide the drug for the clinical trial. But other than that, they conduct a clinical trial in 12 hospitals across Italy and potentially in Europe in patients that have melanoma, and these patients are going to receive surgery. But what we do is ask the patients to delay the surgery by about 8 weeks. We treat them with MDNA11 in combination with a standard of care, which is Bristol Myers' drug called Opdivo or Nivo. And 8 weeks later, we surgically remove the tumor and we get a chance to see if the tumor shrank with MDNA11 plus the checkpoint inhibitor. And if it did, then the question is, why even bother conducting surgery in these patients? Now, in melanoma, it's relatively easy to surgically remove the tumor. But in certain cancers, when you surgically remove it, for instance, if you have a stomach tumor, to remove the stomach has really bad consequences on the patient's quality of life for the rest of their life. If you have anal cancer, if you have rectal cancer or colon cancer or bladder cancer, if you have to remove those organs, the quality of life deteriorates substantially. But if you're able to treat the patient and save the patient from receiving surgery, that would be fantastic, not only in terms of having cured the patient, but also having the patient maintain a good quality of life. So melanoma is our first venture into this early stage disease with the potential of pursuing other earlier stage tumor types that we think could have a huge impact on patient survival and potentially long-term cures where the patient's tumor doesn't come back uh, within weeks or months, which is typically the case with other therapies. So basically, that's where we are. Moving on to sort of the next slide is our second program. This has really been very exciting. There's been a lot of transactions that have taken place. If you look at what we call anti-PD-1 bispecifics, And the reason there is this excitement is that data is showing that when you combine or create a molecule that has two functions, namely the checkpoint inhibitor together with another mechanism, you have a better outcome for these patients. So the top table basically shows that Merck's drug Keytruda and Bristol Myers' drug Opdivo combined annual sales of $42 billion will be going off patent in about 2.5 years. And we feel there is a fantastic opportunity for us now to leverage that information and create much better molecules. So on the extreme right, so if we go back to the previous slide, just to let you know, over the past 12 to 18 months, the transactions in the bispecific space has totaled about $35 billion. The most recent one, and one of the largest one, was the $11.4 billion deal between Takeda and Innovent. And this is a drug which is an anti-PD-1 fused to IL-2. So let us look at this particular program, the anti-PD-1 and IL-2 approach. And that's what we are using at Medicenna with our 113 drug. So going to the next slide, this is what we have is, so what's Medicenna's 113 drug? How is it different from the recent Innovent Takeda transaction? Okay. First and foremost is if you look at that Y-shaped structure, the gray structure on the left on the slide, that's the structure of an anti-PD-1 drug. That's your Opdivo, that's your Keytruda drug, okay? That's what it is. And as I said, that's going off patent. So the good thing here is what Medicenna is doing is taking one of the world's biggest blockbuster drugs and using that to create this bispecific molecule. Second is that green circle that you see there is our IL-2. It's the same IL-2 that you find in MDNA11. MDNA11 has shown single agent activity. It's able to treat patients who failed checkpoint inhibitors. So we are linking the best IL-2 with the best anti-PD-1. Okay. And the third thing we are doing is we're saying we want to have the drug so that it's safe and it localizes at the tumor site. And for that, you can see those pink squares. That's a molecule called IL-13 superkine. And the reason we've put that in is because it localizes the drug. It sort of delivers. It navigates through our bloodstream and takes the drug right to the tumor site. And essentially, the drug, while it is traveling through our bloodstream, it essentially is inactive. So it's not going to cause toxicity. The moment it gets to the tumor, the pink boxes are removed and the drug is switched on. And that's where the drug will stimulate your cancer-fighting immune cells. It does two things. It will bind to the anti-PD-1 receptor on the T cell, which is your cancer-fighting immune cell, and the IL-2 will also bind to the same T cell. So now you've got this super strong, call it the Navy SEALs of your immune system, where you have the best possible immune cells to fight the cancer. And this is where there's a big deal here. And one other thing to let you know is that the IL-13 is actually targeting tumors. And these tumors that have what we call the IL-13 receptor are by far one of the most aggressive tumors. Patients that have the IL-13 receptor have very poor survival outcomes. Patients with pancreatic cancer, colon cancer, brain cancer, etc., breast cancer, or lung cancer, they have high amounts of this particular target known as IL-13 receptor. And every year, 2 million patients are diagnosed with this kind of tumor. And therefore, the market opportunity is massive for 113, where you can deliver this drug right at the tumor site, particularly in cold tumors that don't respond to checkpoints, etc. this is where MDNA-113 will play a big role. And I think we've got a much better version of MDNA-11 or MDNA-113 than Innovent. Innovent's IL-2, as you can see here, has got safety issues. You can see that there is a risk of patients dying because of the IL-2, because it's not been engineered the way MDNA-11 or our IL-2 has been engineered. So that is the one and first foremost thing. The second thing is also when we look at just the efficacy data, Innovent's drug in patients who failed checkpoint inhibitors, you have response rates of only 30%, which is great in patients who have failed checkpoint inhibitors, and which is what we get with MDNA-11 alone. So MDNA-11 alone is just as good as Innovent's anti-PD-1 IL-2 molecule. So this is the big deal that we have, is that first and foremost, the IL-2 we are using has proven to show single agent monotherapy activity in failed patients, in sort of end stage patients. That's one thing. Innovent's IL-2 has not shown, in fact, has never been tested in the clinic. So we don't know how good their IL-2 is, except for the fact that we see there is a high risk of death in these patients. Second, the PD-1 or the anti-PD-1 they use is their own version that's been only approved in China. It's not been approved. It's not like your blockbuster Keytruda or Opdivo. Medicenna's 113 contains the blockbuster Opdivo or Keytruda. So that's the big advantage. So we've got the best of both worlds. We've got the best anti-PD-1, we've got the best IL-2, and we have masked the molecule so it targets and accumulates at the tumor site. So we're basically anchoring our drug right at the tumor site until it does its job. And that's what is unique about our drug, plus the safety. So this is where we are with 113. We are now conducting studies in non-human primates, namely monkeys. We'll have that data in early January. We will hope to share that. And if it shows, the key thing here is for us to demonstrate which of the anti-PD-1s we'll use. Is it going to be Keytruda or Opdivo? We'll select that based on a monkey study. And essentially what we'll be able to share is the fact that we'll be starting the clinical trial with this drug in the second half of next year. And we think that the way the drug is designed, provides us so many more advantages than the other competitors out there. Okay?
Fahar.
Yeah.
Sorry, just um, connecting 11 with 113. They're both using the same
I,
IL-2 superkine, correct? They're
yes.
just attached to different other molecules and being targeted in different systems in the body, if that's
Correct. So MDNA-11 is basically linked to albumin, so it accumulates in the tumor draining lymph nodes, etc.
yeah.
Uh, this IL-2 in MDNA-113, it's linked to the checkpoint inhibitor, anti-PD-1. Okay, so that's the difference. And to localize the drug and make it accumulate at the tumor site, we're using an IL-13 superkine. So that is very, very specific. It will only bind to tumor cells that have this particular target. Okay.
So what I guess I'm getting at, when you get data that the your IL-2 superkine works using the MDNA 11, that also provides evidence. Well, it works here, and then
Yes,
combined with it can hopefully then work out. So the data from one supports to some extent data in the other program. Is that correct?
absolutely. And that's
Okay.
what we've demonstrated that our IL-2 on its own works.
Yeah.
Uh, and there's plenty of evidence. Obviously, we all know the anti-PD-1s are for Keytruda or Pembro. Those are blockbuster drugs, so we know those work as well. So basically, taking the best of both worlds and link them together. So finally, I think, we don't have too much time, so I won't spend much time here on Bizaxofusp, simply that, we want to give an opportunity for Q&A. So if there are any questions, we can address it. But suffice it to say, this is a program where we are looking to partner this asset. This is a drug to treat patients with recurrent glioblastoma. And just for a bit of background, GBM is by far, like pancreatic cancer, one of the most aggressive cancers out there. Patients typically get, when they're diagnosed, about 80% of the patients will get surgery. They will then get radiation with a drug called Timodar, a chemotherapy drug, and this will be like a 6 weeks of treatment. And then patients get this high dose of this chemotherapy until the tumor comes back, which unfortunately happens in the majority of patients. So GBM is, the tumor type. I think from Canadian perspective, we know Gord Downey. He passed away because of GBM. In the US, we had Bo Biden, son of the former president, succumb to GBM. We had uh, also one of the Kennedys, as well as Senator John McCain, all succumbing to GBM. And there's really been nothing out there for the past 30 years, particularly when the tumor comes back. And when the tumor is recurrent, that means the tumor has relapsed. Those patients have much more deeper, more aggressive tumors, and majority of these patients will pass away in about 6 to 9 months. So the key thing here, just to give you a message here, is the drug that was approved 30 years ago was Timodar, and this is still the standard of care in newly diagnosed patients who are first diagnosed with brain cancer. Timodar has improved survival by 10 weeks and became a billion-dollar blockbuster for Merck. We, on the other hand, are looking at patients who have failed Timodar, have failed surgery, have failed radiation. The tumors come back. It's now deeper in the brain. And our data, if we show the next slide, will show that we've been able to demonstrate that not only have we increased survival with bizaxofusp, we are improving survival not by 10 weeks, but we're improving survival by several months. It's about 7 months, nearly doubling the survival in these patients. So really encouraging data. We'll be publishing some updated results from the clinical trial. We are partnering this asset. I know we've been trying to partner this asset, but they've been really bad data from previous attempts by other companies trying to develop a drug for GBM. So we think now there's been a bit of interest in this GBM space. We're seeing a lot of inbound interest. We're hoping that we'll partner this program sometime next year. And not only for GBM or recurrent GBM, we feel there's opportunity for tumors in the brain that spread from the breast, from the colon, from the kidney, because there's a much bigger market opportunity. So potentially a huge opportunity for GBM. And our current focus and the spend of the company's current cash resources is on MDNA 11 and 113. We have enough cash to take us into Q3 next year. And we have, as I mentioned, RA Capital as the largest institutional shareholder. Insiders own 22% of its outstanding shares, so there is really skin in the game. And we have analyst coverage by various banks out there, the most recent one based out of New York by Lucid Capital. So key highlights, I would say, in the coming weeks, we'll be presenting data on 10th of December on MDNA 11, the monotherapy and combination data. We are planning to complete enrollment in this clinical trial, as soon as we possibly can. It's sort of heading nicely. As I said, we've enrolled about 120 patients so far. So these are the key data readouts coming up in the next few weeks. We also will have data by early January on our non-human primate study with the bispecific MDNA113. And we continue to advance Bizaxo in terms of trying to partner that asset. So with that, I'll stop and take questions.
Fahar, thank you. That was, I feel a lot smarter now. I feel like I know a bunch more, so I appreciate that. I'm just curious, since you have it on the screen here, you're releasing the data on December the 10th. Is that
Yeah.
in conjunction? Is that the ASH conference, or is that are you presenting data at a conference where you've sort of booked the time in that, or can you
Yeah.
what's the rationale for December 10th?
So December 10th is consistent with the conference called the European Society of Medical Oncologists. This
Okay.
ESMO meeting taking place. It's focused on immuno-oncology taking place in London in UK. And that's when we'll be presenting our data at that conference. So we'll share that pretty much on the same day.
On the MDNA 11, you've been doing end stage cancers. You focused on that, and then with the melanoma, that is you're focusing on earlier stage stuff. Is there a reason you started at the end stage? Is it easier to get regulatory approval since they don't have many options, or what was the rationale for that end stage strategy?
A really good question here. And I think I appreciate that because I failed to mention this. But one of the reasons that you would look at end stage patients is there is a good possibility, particularly in the tumor types that we are looking at, that the FDA would allow you to conduct a phase 2 trial, and that would be enough to get you approval based on about data from about 50 to 100 patients. So that is one of the reasons, and that's one of the approaches that we are pursuing with MDNA 11 in end-stage patients. Although we have, conducted the phase 1/2 trial, we have demonstrated that the drug works on its own or in combination and in patients who have failed standard of care. So these patients generally do not have any other treatment options. And the only thing they can do is enter a clinical trial. So the FDA and other regulators, including the Europeans, will allow you to get the drug approved based on a phase 2 trial. So that's the way to get into the market quicker and faster and less expensive.
The data we're expecting on December 10th, is that powered enough that that could trigger the accelerated approval, or would you
No.
need an additional study
Yeah, we would need
to
to do
hit
a
that?
we would need to do a small phase 2 study. Yeah, so we
Okay.
need more patients. So anywhere typically from 50 to 100 patients. It could be as many as just 70 patients to get the data readout to get approval. So it's relatively small compared to, let's say, melanoma in newly diagnosed patients that might be hundreds of patients. So that's much faster, quicker, and quickly you could transition into first-line therapy after that.
And given the data that comes out, if it's positive enough, do you then take that to the FDA and they say, hey, this looks potential that we could do a smaller pivotal trial for end stage? And then do you kind of get pre-approval for them? Then, yeah, this structure it
Yeah.
like this. And if you get past certain thresholds, this could trigger the accelerated approval.
Correct, exactly. So basically, once we have this data early next year, the plan is to meet with the FDA, provide our data, and get their feedback with respect to a potential pathway for accelerated approval. And then based on that, we would then design the study to meet the FDA guidelines and pursue a small phase 2 clinical trial, either our own or in partnership with a bigger pharma.
All right. And I guess I just want to go back to broader on the MDNA 11 because it triggers immune system. It just dawned on me that because you're targeting a lot of drug companies I talk to, they have one drug targeting one very specific indication. You're like all sorts of tumors, melanoma, pancreatic. Is that because it is like an immunotherapy where it just gets the body like the soldiers trained and so forth? And then I guess there was one chart you showed that like as once you each knock down each of these target, it just potentially creates a much bigger market for you or many markets for you to go after.
Correct.
Is that a good way to think of it?
Very good way of thinking of it. It's very much like Keytruda. For instance, Keytruda today, you know, is used to treat a couple of dozen tumor types. So you can imagine there's sort of a huge opportunity. Although the first approval was for melanoma, it's now essentially being used for 20, 30 different tumor types. So that's the same approach here. By Keytruda or Opdivo, those don't attack the tumor. They basically what they do is they remove the brakes from the immune system so that the immune system can now function because the tumor is clever. It sort of puts a brake on the immune system so that the immune system does not recognize the tumor. The immune system is neutralized. But by using Pembro, they basically restore the sort of remove the brakes on the immune system so you can attack. What MDNA11 does is basically takes the immune system and trains the immune system to fight the cancer. And I think that's where the big difference is uh, in terms of being able to use the drug for many more tumor types rather than just one particular tumor type.
Okay, we've got a bunch of questions from the audience. Fahar, can you talk about the interest in partnering activities with Medicenna and all the recent conferences Medicenna has attended?
Yes, of course. I mean, the thing is, particularly with MDNA 55, there's a renewed interest with the GBM or the glioblastoma program. But more interestingly is the recent transaction of Takeda with Innovent. As you know, in the past, the IL-2 space had multiple multi-billion dollar transactions that took place. Unfortunately, the IL-2 programs that they were developing were not showing activity, and therefore the interest in the IL-2 space diminished. But now, again, with the recent deal with Innovent, the interest in the IL-2 space is reinvigorated. The data readouts that we will share in the coming weeks and months will be based on MDNA11, but also MDNA113. We think, and we've seen that a lot of interest, a lot of inbound interest coming into Medicenna without us soliciting any meetings. We've been getting some encouraging feedback on the data. So really excited about what the sort of the upcoming milestones are, but also the interest that we are generating with our platform.
I'm going to stick to the sort of the corporate side of things here. You mentioned recently big pharma in M&A buying spree, likely driven by cliff dynamics.
Right.
Uh, what stage does Medicenna need to be at, I guess, for the various programs to be an M&A target? And I just want to break that out a little further. With the different programs, there's M&A potential, but there's also licensing.
Correct.
Like obviously the GBM indication that's ready and packaged up for it. I guess you're getting a little more data. But for each of the MDNA 11 and the 113, like is MDNA 11 have enough data, or obviously they're going to wait for this upcoming one here? I've said a lot, and sort of please
Yeah.
respond to that general theme.
Yeah, so no, certainly. I mean, the key thing here is, as you mentioned, there could be partnership transactions, it could be licensing, very much like what Innovent did. It's was an $11.4 billion deal with about 1.2 billion upfront. So, in a sense, it could very well be a meaningful partnership transaction and not necessarily an M&A transaction. It could be both. I mean, it could be M&A for the program, but MDNA 55 or Bizaxo could be partnered. So there's sort of different ways of structuring that. The key thing here is data-wise, the data that we are generating with MDNA 11 so far, the feedback generally is from a pharma company perspective, typically, data in different tumor types would be ideal to have data from about 20 patients for each tumor type, which we are approaching in some tumor types. So we are, continuing to build that database from MDNA 11. With MDNA 113, it could be. I mean, the recent transaction was with Innovent's drug already in the clinic, although their clinical trial has been conducted all in China. They're sort of starting next year a clinical trial that'll be global, so it'll be enrolling patients in US and so on. But I think, as we've done in the past, we'll be studying the clinical trial right away with MDNA 113 globally, starting in the territories that we've used with MDNA 11 in the past. The question is, will they wait for clinical data to transact with Medicenna on 113? Or is data from non-human primates going to be sufficient enough together with our preclinical data for them to get comfort around the fact that the drug is safe, it has the right profile with respect to its pharmacokinetics, pharmacodynamics, etc. Because the big negative about Innovent's drug is that it is toxic. And just to let you know, it wasn't Takeda only bidding. There were other companies bidding as well. So there was a competitive process going on. We don't know who the others were, but we know they were. So there is certainly interest in other big pharmas to acquire a anti-PD-1 IL-2 program. But I think the key thing here is the differentiation will be the fact that we have an active IL-2, and best-in-class checkpoint inhibitor, and a safe molecule. So that's the key thing.
All right. Is the enrollment completed in both mono and combo, or is it still ongoing?
Still ongoing. We are continuing to enroll until the end of this year. We'll be looking at some of the things that, as we are contemplating a phase two registration trial, the FDA generally likes to have something called Project Optimus. They want to be sure of what dose we will implement in a phase two registration trial. So we may extend that to add another dozen or so patients. But that's basically been already incorporated in our sort of runway calculations.
With that potential or that expected potential small phase two study for the FDA for an accelerated pathway, just ballpark, like roughly, would that be a 1 year or a 2 year? Can you give some indication of how long that process would take?
Yeah, it depends on the tumor type. So, for instance, melanoma, endometrial cancer, those are much faster to recruit than patients with, let's say, MSI high tumors. TMB high tumors, again, much faster to recruit. So it depends on the tumor type. It could be as short as a year or as long as 2 years, depending on the tumor type. So that's it. It sort of ranges. Yeah.
All right. What objective response rate or ORR percentage is considered very good for the upcoming MDNA 11 drug? What would be good results there?
Yeah, so really the threshold or the benchmarks that we want to sort of achieve, I think with endometrial cancer, for instance, the response rates there are very poor in this patient population. It's as low as 5 to 8%. So if you get response rates well above that in the sort of 15-20% range, that'd be fantastic. MSI high, TMB high, again, these are patients who have failed checkpoint inhibitors. The benchmarks we've been told is going to be about in the 20% range. With melanoma, 30% would be fantastic as well. So those are the sort of the range. Anywhere, depending on endometrial cancer being the lowest bar, to melanoma being the highest bar.
On December 10th, when you present, will you be presenting a poster or will you be making a presentation?
This will be a poster presentation
All right.
by the investigator. So it's the one of the hospitals that's treating patients will be doing the poster presentation.
What has Merck's, your partner's feedback been on MDNA 11 combination data so far? Can you comment on that?
Yeah, they obviously are encouraged with the data so far. We continue to interact with them every quarter. We certainly cannot disclose publicly what discussions we're having with them, but suffice it to say that they are encouraged with both monotherapy and combination data of MDNA11.
Is there benefit or potential significance that Bristol Myers is supplying the checkpoints for the Italian study versus Merck, who's supplying the Keytruda for your current MDNA studies? The fact that you're working with two large pharmas here, should we read anything into that? Obviously they know who you are.
Yeah, I mean, the thing is this, it's obviously we want to use the drug that is currently standard of care for neoadjuvant purposes. In melanoma, the standard of care is the Bristol Myers drug, which is EP and Nivo. So this is sort of Opdivo is the standard of care in neoadjuvant setting. Pembro is not. So that's why we are working with Bristol Myers on that. With the other tumor types that we've been talking about, like MSI high, the endometrial cancer, the TMB high, etc., multiple and melanoma, the multiple checkpoints have been approved, including Pembro. So basically hedging our bets, I would say, having both trying the drug with both different drugs, both checkpoint inhibitors, including 113. We have a version of 113 that contains Bristol Myers' Opdivo. We have a version of anti-PD-1, which is a Keytruda, Merck's drug. We'll be testing both of them in non-human primates, and we'll pick the best one of those two.
You said you have cash runway right now until Q3 of 2026. There's a question here on when will you raise cash? I guess that's a little bit based on, and just discuss these different, there's so many different paths that could happen. How good the data is, and then does that create a partnering or a licensing deal and so forth. Or you may just raise equity on the back of good data. Can you just talk about the different potential funding paths you have ahead of you?
Yeah, it could be any one of them. So all options are open in terms of, they could be inbound interest. Just to let you know, we had, for instance, RA Capital on the back of good data last year. This basically came in with a commitment to a reasonably sized $20 million check. So, the key thing here is, uh, we could do that if that occurred. The potential for partnerships, potential for collaboration, etc. Those could be, again, discussions that would be in the open in terms of open to that kind of deal structure as well, and or do a equity raise on the back of more data from the MDNA 113 program as well. So it's sort of no particular timetable per se, but certainly if we are aggressively going to pursue a phase 2 registration trial, yes, we will need additional capital. And the question is, where does that come from? And that sort of could be inbound interest, could be something we pursue on our own with the banks, or it could be a partnership deal, which could bring non-dilutive capital in the company.
And you can weigh them all against each other depending on how strong the share price is. You could
Yeah,
do equity that would maybe be better than a partnership, or the partnership could be very advantageous, or do a combination thereof.
correct. Agreed. Agreed. Yeah, the more clinical data we have, the better deal structure we can have in a partnership setting.
Do you envision MDNA 11 and MDNA 113 working together over time? Is the idea that MDNA 11 restores immune architecture and 113 amplifies the response?
Yes, really good question. And that's something exactly what we are also studying right now is certainly sequencing the treatment approach. MDNA11, as I mentioned to you, is generating on its own, boosting the population of immune cells very substantially. MDNA11 is generating your sort of call it high quality cancer fighting immune cells, call them the Navy SEALs. And as you do that, the question is, how can you further push the immune system. And this is where MDNA 113 comes in play. So once you've built up your army, let's put it this way, you've got enough soldiers now, and now you want those soldiers to be very targeted, very specific, precise, and execute their mission. That's where 113 comes in. So it sort of makes obvious choice instead of combining MDNA 11 with a checkpoint. Why not combine MDNA 11 with 113?
What's the difference between the current combination trial MDNA 11 plus Keytruda versus MDNA 113?
Well, that's a good question. Again, I think the key thing here is the MDNA 11 purpose is different. It's basically to build up the immune cell population. And the second thing is the Keytruda is there to simply release the brakes on the immune system so the immune system can go working at it. Now, with that approach, when you're injecting two drugs separately, the IL-2 is going to stimulate your immune cells, and the anti-PD-1 is going to release the brakes on immune cells. Does it do it on the same cells? Probably not. So what happens is when you administer MDNA 11 on its own and Keytruda on its own, two separate injections, the probability that both MDNA 11 and Keytruda will attach to the same immune cell is low. Whereas if you have 113, the way it's designed, the two molecules are connected together. Once it binds to the IL-2 and binds to the anti-PD-1, that effect is occurring on the same T cell, the same immune cell. So now you're creating a different kind of immune cell. This is your sort of called it Navy SEALs of your immune cells. And that's what it is. So that's basically when I said you would probably treat patients with MDNA 11 first to really build up the T cell population, and then you treat patients with 113. So now all those T cells or your soldiers, instead of only some being your Navy SEALs, all of them are your Navy SEALs. All right. So that's basically the difference.
All right. And then one final question here, and then we really will wrap it up. Are you planning any orphan or breakthrough therapy designation for any of this from FDA?
Yeah, so certain indications, I think if you look at patients who have failed, let's say checkpoint inhibitors, that being the case with melanoma, for instance, that being the case for endometrial cancer, for instance. So those are potential areas where you could seek out orphan drug, fast track, etc. designations with the data that we have on hand. So yes, certainly we'll be looking at that as well.
All right, Fahar, we should wrap it up now. Thank you very much. Any final thoughts or ideas to leave viewers with as we wrap it up here?
Yeah, I think it's really important to say one thing is that the IL-2 that we have is really the core of a lot of uh, you know ability that it has to really boost the immune system. I think it's highly differentiated with so many other IL-2s being pursued. The way we've engineered the molecule uh, addresses so many key issues that are not being addressed by other IL-2s or other cytokines for that matter. And the fact that we are sort of leveraging our drug in terms of, using the sort of the uh, albumin to localize it at the lymph nodes, that is really important because you need a versatile lymph node in and around the tumor. And albumin allows us to build that and repair that lymph node so that you have a productive lymph node producing high quality immune cells. And that is, again, something that's not being done by others. And the third thing is that it's a really strong prerequisite in the sense that I believe that MDNA 11 will become a prerequisite for other immunotherapies. It could be checkpoint inhibitors, it could be oncolytic viruses, it could be cell-based therapies, it could be antibody drug conjugates. We think that all those treatment modalities, if helped by MDNA 11, will do much better because you'll be able to generate what we call memory immune cells. And these memory immune cells then behave like almost like your own personalized vaccine that prevents the tumor from coming back, as we've seen in a couple of patients already, despite the fact these were end-stage patients, to see the tumors disappear and not show up for several months or now a couple of years with the pancreatic cancer patient. So that's, I think, the long-term goal is to destroy the tumor, but hope that the tumor doesn't come back. And that's basically the aspiration is essentially to do those two things. Okay.
Fahar, thank you. That's leaving it with a big ambition there, big opportunities. So thank you very much. I really appreciate your time. Very informative, very exciting. We got lots of potential catalysts here coming up over the next few months. So that's very exciting and good to see. Thank you.
So, okay, take care.
You too.
Okay, bye-bye.